So I'm Paul. I work from Necker Hospital currently, and today I'll briefly talk to you about our work in the last 18 months about the genetic predisposition to severe COVID-19, and in addition, the autoimmune predisposition to severe COVID-19 via the autoantibodies against type 1 interferons. So as a brief introduction, we all know these images of patients with critical COVID-19 pneumonia, very severe lung affectation following SARS-CoV-2 infection. Despite these images being now infamous, the prevalence of such severe disease is actually relatively low, and most infected individuals are asymptomatic or very mildly symptomatic. So the question that we tried to tackle here and that our lab led by Jean-Laurent Casanova has been tackling for more than 25 years is how do we explain such differences between two individuals or more individuals who've been infected with the same virus or other pathogen, but who develop a very different clinical spectrum of a disease. So focusing on COVID-19, there's a few risk factors that are known at the epidemiological level. The first one is the age, and the risk of severe COVID rises with age, with a risk doubling every 5 years. The second risk factor is being a man, and then there's a multiplicity of other comorbidities that have been associated with severe COVID, for example, diabetes, obesity, and so forth, but their odds ratio, their relative risk is relatively low, less than 2. And even within one age group of individuals with the same comorbidities, there remains a very large spectrum of clinical disease. And for example, centenarians have handled well the virus. And on the other hand, young women have unfortunately died of it. And so the question we posed was, what are the causes of life-threatening COVID-19? So to answer this, Jean-Laurent Casanova teamed up with Helen Su at the NIH to create a worldwide consortium to recruit as many patients as possible with either asymptomatic infection or very severe disease patients that were in the ICU. And as of today, we recruited more than 10,000 people from, as you can see, all over the world and from more than 400 hospitals who participate in this study. As a first, let's say, area of focus, we decided early last year to focus on type 1 interferons. So type 1 interferons are a quite large family that was actually discovered a while ago in the 1950s, and that basically all bind to the same receptors, IFNR1 and IFNR2, shown here, and triggering, after a signaling cascade, a very potent antiviral response. And so it's been shown since the 1950s until today that type 1 interferons are really the first barrier against any viral infection. What is surprising is that there's actually 17 types of type 1 interferons. It's a very large family, but they all look very much alike, especially the 13 interferon alphas and interferon omega. And we'll also discuss a bit later interferon beta, which is slightly different at the structural side. And so what's been known after discoveries by our labs and other groups is that patients who have deficiencies in type 1 interferons because of genetic inborn errors of immunity are actually susceptible to very severe viral diseases. For example, adverse reactions following MMR vaccination, yellow fever vaccination, herpes simplex encephalitis, or flu. So we decided to focus our attention last year on looking for inborn errors of type 1 interferon immunity in patients with life-threatening COVID. And so at the time, we had about 600 individuals with severe COVID-19 and close to 400 with asymptomatic or mild infection, for which we sequenced the exome or the genome, and we looked for damaging mutations in the genes that are shown here in the lower left that are involved in the production or the response to type 1 interferon. Very surprisingly, we found that 2 to 3% of the individuals that we tested had actually loss of function mutations in one of these genes. Even more surprisingly, we found 4 patients who had either autosomal recessive complete IFNR1 deficiency, meaning they do not have the receptor to type 1 interferons and they're thus completely unresponsive, to any of the 17 type 1 interferons, and 2 other patients with autosomal recessive IRF7 deficiency, who, because of this deficiency, have a defect in the production of type 1 interferons. Why was it surprising for us? Well, because previously, the patients with either IFNR1 or IRF7 deficiency were only known to be young children. In any case, from this first discovery, we wondered if other inborn errors of type 1 interferon immunity could underlie life-threatening COVID-19, not only in males and females, but specifically in males. And so a few months ago, Takaki Asano from our New York lab published the discovery that there was a very strong enrichment of damaging mutations in the gene called TLR7, which is located on the X chromosome, in patients with critical COVID-19. And overall, you can see here the pedigrees of the 14 families that were in which we found these loss of function mutations. Overall, more than 1% of men below 60 years old who suffered from critical COVID-19 had TLR7 deficiency. And so TLR7, I'll show it a bit later, is involved as IRF7 in the production of type 1 interferons. So overall, from these genetic studies, it looks like that in a small proportion of young patients, inborn errors of type 1 interferon immunity can be the cause of life-threatening COVID-19. This being said, we found very few elderly patients with these mutations. And also, although we explained 3 to 4%, this was still a small percentage. So we wondered if we could find more frequent causes that could explain severe COVID-19, especially in the elderly individuals. And so for that, we turned to autoantibodies against type 1 interferons. So why did we think of this? Well, autoantibodies against cytokines have been known since about 20 years ago to underlie other severe infectious diseases. For example, tuberculosis can be caused by autoantibodies targeting another family of type of interferons, type 2 interferon. And for autoantibodies targeting type 1 interferon, the antiviral ones, they have actually been known since the 1980s, and this is very surprising for us because they were shown to be existing in patients who were treated with interferon alpha for hepatitis, for example, in women with lupus, and in all patients with a rare genetic disease that is a really severe autoimmune disease called APS1 or APOSET. In all these patients, they were thought to be clinically silent. Nevertheless, when we go back again in the 1980s, there was one patient who was described in 1984 to have severe varicella and autoantibodies against type 1 interferons. And then in 2015, several patients with hypermorphic RAG1 mutations were found to have autoantibodies against type 1 interferons and viral infections. And finally, unfortunately, COVID-19 arrived, and we heard about 3 APS1 patients, which we knew had these autoantibodies, who were young patients and suffered, unfortunately, from critical COVID-19. So based on all these observations and knowing that type 1 interferons were crucial to fight against viruses, we wondered if they could not be neutralized by autoantibodies and thus, in that case, cause severe COVID-19. And so to answer this question, we recruited, again, an international cohort of about almost 1,000 individuals with severe COVID-19. And as a control group, we looked in patients with asymptomatic infection. And so here, very surprisingly, we found that more than 10% of individuals with critical COVID-19 had high titers of neutralizing autoantibodies against interferon alpha-2 and or interferon omega. On the other hand, in the group of the asymptomatic controls, at the time, we found that none of them had these autoantibodies that could block the interferons. And finally, as a second control group, we looked in a healthy population whose samples had been collected before SARS-CoV-2 existed. And in that population, we found that 0.3% of individuals had these autoantibodies, suggesting they were pre-existing. To further confirm this, we tested as many patients as we could, and in all the patients we tested, for which we had samples before the infection, we found that the autoantibodies were already present. Two other interesting observations were that the fact that in most patients, the autoantibodies were present in men, in about 95% of patients, and more than half of our cohort of patients with autoantibodies were over the age of 65. So potentially, these autoantibodies against type 1 interferons could explain why there's an increased risk when you're a man, and especially when there's an increased risk with age. These findings were reproduced all over the world, as you can see here, and we tried to further confirm that these autoantibodies were not just associated with the disease, but were the cause of the severe viral disease. And so for this, we teamed up with Charlie Rice's team at the Rockefeller University, and they did a very elegant experiment. So here what they did was to infect control cells with SARS-CoV-2 and look at viral replication in presence of interferon alpha. And so when you do this, in addition to adding plasma samples from healthy controls, you see here that there's no viral replication at all. On the other hand, when you add plasma samples from patients with autoantibodies neutralizing the interferons, well, there's a very high viral load, even when we dilute the plasma samples 10,000 times or more. So overall, these autoantibodies could block the protective effect of interferon against SARS-CoV-2. So overall, from this first study, we learned that at least 10% of patients with severe COVID had autoantibodies pre-existing the disease that could block the protective effect of type 1 interferons against SARS-CoV-2, leading to very high viral replication. This is important because these autoantibodies, like most autoantibodies, can be easily found by an ELISA, and in the patients who have them, they can be treated with interferon beta, monoclonal antibodies, or if it's too late and the patient is already severe, we can try plasmapheresis. In addition to this first description, we wondered if we had maybe missed some patients, and if more patients could have autoantibodies neutralizing lower concentrations of type 1 interferons. And for this, we set up a new, more sensitive assay to look at the neutralization. And here we found that not 10%, but it was closer to 15% of a larger cohort of patients with critical COVID-19 who had autoantibodies neutralizing these lower doses of type 1 interferons. We also looked in a group of patients with severe COVID-19, hospitalized but not in the ICU, and found that about 8% of them had the autoantibodies. And at this lower dose, about 1% of the controls had the autoantibodies, meaning that the presence of the autoantibodies gives a very high risk of having the disease, but not in all cases. There is incomplete penetrance. So as I mentioned previously, there's 17 type 1 interferons, and here we looked if these autoantibodies could neutralize the 13 interferon alphas. And as you can see here, this is the case for the patients who have autoantibodies against interferon alpha 2, but this is not the case for patients who have autoantibodies against interferon omega only. Furthermore, we also found that, surprisingly, at least 1% of patients with critical COVID had autoantibodies blocking interferon beta, suggesting that interferon beta deficiency alone can lead to severe COVID-19. And as you can see here, we calculated the relative risk of having severe COVID-19 when you have these autoantibodies. And when comparing to the other risk factors that have been described, the odds ratio here are extremely high, more than 50 for patients who have both autoantibodies against interferon alpha 2 and interferon omega. And the last observation was the fact that these autoantibodies increase with age in patients with severe COVID-19. And so we wondered, what is the prevalence based on age in the general population? So for that, we recruited more than 34,000 individuals and tested them all with our neutralization assay. And of note, these samples were collected before SARS-CoV-2 arrived, so they were all collected in 2017. And here, strikingly, we found that in uninfected individuals, the prevalence of autoantibodies increased very significantly with age, especially after the age of 70. So potentially this is an explanation of why elderly patients are at higher risk of critical COVID-19. So overall, either from the genetic side of patients, young patients with inborn errors of immunity, or the autoimmune copy of these genetic defects, autoantibodies against type 1 interferons in all ages, but especially in the elderly. The common mechanism underlying critical COVID-19 is a deficiency of type 1 interferons. So, as I mentioned previously, we found autoantibodies in more than 20% of patients older than 80, and in more than 20% of patients who died of COVID, whatever the age. Surprisingly, some patients had autoantibodies against interferon beta only, and the prevalence of these autoantibodies increased with age. So type 1 interferons look like they're clearly crucial for the protective immunity against SARS-CoV-2, and severe COVID can result from a deficiency either because of a genetic cause or, more frequently, an autoimmune copy of this genetic cause. This is, I think, high encouragement to look for these autoantibodies and for the genetic defects and to treat these patients early. And the good news is that vaccination works for these patients. They are well protected, and so they should all be vaccinated as early as possible. And with that, I'd like to thank, as you can imagine, these studies involving thousands of patients involve a lot of friends and collaborators all over the world. And I'd like to thank all the physicians that participated, the patients and their families who accepted to send us samples, both branches of our labs in Paris and New York, Jean-Laurent Casanova and Laurent Abel, who lead the lab, the autoantibody team that you see here underlined, and then all our collaborators who have helped with these studies, either by performing experiments or sending samples. Thank you very much, and I'm happy to take any questions.
Thank you, Paul. That was outstanding. There is a burning question that I will forward to you right away from the audience. Are there data to support an inverse conclusion, meaning that a better type 1 interferon response would be associated with the protective effect, which is probably more tricky to tackle? Do you have any evidence for gain-of-function mutations or?
Yeah, this is a very interesting question. So we've tried to answer it. We actually worked with Yannick Crow from Imagine Institute, and we tried to see if patients with interferonopathies who have very high levels of type 1 interferons had been infected with COVID. And so far, I don't know if they had been infected or not because they were very much protected by their families. So we haven't heard of any patients with excessive type 1 interferons who have suffered from severe COVID, but it's possible that they have not been infected. But I think that the study of patients with high interferon response would be extremely helpful for us too.
Thank you. I have one quick question. I mean, that's a dermatology question. Any of these, two quick questions. One, for the patients with the deficiency, complete deficiency of expression of IFNR1 and 2, right? Was any of these patients developing any other viral infection? Because that would be not necessarily for the autoantibodies developing patients. One can expect that a more virulent, more aggressive agent would reveal the deficiency, but for these cases, we would expect that some clinical events before.
So I absolutely agree with you, and this was very surprising, but we did not find previous viral infection in these patients with complete deficiency that were actually adults. So, and we performed a very large antibody screening for other viruses, and they had been infected with other viruses. For example, flu, they had been vaccinated, but did not suffer from severe disease. So there's clearly things we do not understand, and there is incomplete penetrance for less virulent viruses, yeah.
Thank you. And any patients with psoriasis as a comorbidity in these patients because interferon are pathogenic in psoriasis?
Absolutely. So within our initial cohort of 100 positive patients with autoantibodies, 2 had psoriasis as an associated autoimmune condition. Because it was still a small number, we cannot conclude anything, but now we're trying to look at it from two ways. We're trying to collect the information of all the autoantibody positive patients, and we're also trying to look in patients who have other autoimmune diseases if they have autoantibodies type 1 interferon. So if you, I would love to test if you have cohorts of patients with psoriasis for which you have samples. I think it would be incredibly interesting to look for anti-interferon autoantibodies.
Harry, one question, if I may, here at the end. I've been aware of, like, there have been use of JAK inhibitors to treat COVID, and yeah. Given the beautiful work and the schematic and the role of type 1 interferons in the process, how do the JAK inhibitors kind of fit in?
Yeah, so that's a very important question too. So the JAK inhibitors, I'm not a super fan in COVID-19 because, as you mentioned, it's kind of against what we found as a risk of severe COVID-19. Nevertheless, like steroids, I think that when they're given at a very late stage, it's probably protective against the overt inflammatory response. So our model is that if you have a deficiency in type 1 interferons, you have a very high viral replication that leads to hyperinflammation that is then deleterious. And then when you treat with steroids or JAK inhibitors, it's usually quite late. The patient is already in the ICU. And so at that stage, the virus is less of the problem than the overinflammation.
Makes sense, like a bimodal, kind of biphasic.
Exactly, a biphasic disease, exactly, yeah.
That's a very important question. Thank you, Johan. And that was in practice with patients under JAK inhibitors and clinical trials during this period. We had some very fruitful exchanges with Jean-Laurent Casanova, who was incredibly helpful in this moving territory. So that's really great. Thank you.